Just like to start by thanking the organisers for giving us a chance to actually present today and show you some exciting data and investment opportunity around the company Ryboquin. I should just give some very brief background to who we are. The company is Ryboquin. We're developing gene therapy based oncology products, and as part of that we currently license in a technology called LipTide, which is a delivery vehicle for gene therapy, and we're in the process of merging with the company that we licensed this from to develop a new company that will focus both on products and a delivery platform for nucleic acids, which obviously has been one of the sort of holy grails of gene therapy, has been the delivery problem. Okay, so we are aiming to build a company to commercialise the nucleic acid delivery platform, and the way we're going to do this is with our own in-house product, which is due to go into clinical trials in April 2018, that will validate the platform, adding value to the platform and showing the platform can deliver, all being well. And a suitable sort of model might be our Newman in the States, which has a market cap of around 4.2 billion currently at the moment, and they also develop non-viral gene delivery platforms. Now, we'll talk a bit more about those later and how we differentiate from them. So the technology itself, the unique inventive step here, the pointer's not working, is the peptide element, this delivery vehicle. Most non-viral delivery vehicles are just made up of liposome, and they're non-targeted, and for oncology purposes, they just rely on the leaky vasculature around the tumour to leak out and hopefully bump into the cancer cells and get taken up. The inventor here, professor at UCL, he actually created a peptide nanoparticle originally, just containing peptide. The peptide had two functions. One was to bind the payload and allow self-assembly, and the second was you could actually have a targeting sequence to target specific different cell types. When he then tried this in studies, unfortunately, the particle did not dissociate when it entered the cell. So to overcome this, he added a liposomal component as well to it to mix the two together, allowing it to dissociate once it's inside the cell, releasing the payload. One of the key things about this is actually the flexibility of the payload. It can be anything from a small siRNA right up to 100 kilobase artificial chromosome. So, what differentiates LipTide from some of the other delivery vehicles out there is the ability to customise it, essentially. Choice of delivery route. We have excellent in vivo data, both using this as an infusion, a tail vein injection, or as a nebulised aerosol delivery, and we have data in mini pigs on that, showing successful delivery and gene expression in lung epithelial cells. Like many other nanoparticles, the physical properties can be adjusted. You can flip the charge, it can be cationic or anionic. You can change the size as well to tailor it to the where you're trying to target. And the key element, as I mentioned already, is the idea of this peptide sequence, which you can target to different receptors. For a lot of the oncology studies I'll show briefly, we're targeting integrin receptors, which are known to be overexpressed on many tumour types. For the airway delivery, we're targeting a receptor called ICAM1, which has been shown to be specifically overexpressed on airway epithelial cells. And finally is the payload flexibility, which also sets us apart from many of the other products here, the products around. So it's targeted gene engineering. So the first in-house product we're developing, which we believe in its own right is a very important product, is a tumour apoptosis enhancer. About 50% of solid tumours have mutations or inactivations in the p53 cell death pathway. So this means when challenged with chemotherapy, radiation, or even potentially newer, more targeted agents, which work by causing cell death, you'll not get cell death because the cell is resistant to it. Those cell death pathways are missing. What we aim to do is upregulate a protein called p73, which can replace the mutated or inactive p53 and resensitise cancer cells to therapy. And as part of this development programme, as I said, we aim to be first in man in April 2018, we will look to validate and get demonstration of delivery as well. So developing the product and validating the platform at the same time. I'm now, hopefully, if it's going to work, to show a short animation to explain the science behind our first product, which we call ECP102. So in normal cells and cancer cells which have active p53, the p53 protein, shown here in pink, binds to DNA, initiates what's called the apoptotic cascade, which is cell death. So in response to challenge such as chemotherapy, the cancer cells apoptose and die, and treatment hopefully works. However, in about 50% of cancers, especially colon cancer, ovarian cancer, p53 is mutated. Some of the most common mutations in the DNA binding domain. It can't bind to DNA. You don't therefore get apoptosis. And the cancer cells go on, divide, metastasize, and obviously eventually kill the patient. This is p73, which can take the place of p53. Acts in a very similar way. This here is the mRNA for a gene called itch, and this is the gene we're looking to silence with this delivery vehicle. This is the itch ligase here. Itch ligase is a member of the ubiquitin ligase family, of which is about 600, and they're highly specific targets. And what ubiquitin ligase does, it adds ubiquitin groups onto the p73 protein. Once it has added 4 of these groups to p73, p73 is taken away to the proteasome, where it's broken down and digested. And this is the reason why in most of your cells, p73 expression is at a low level, as you would want it, because you wouldn't want all your cells in your body exquisitely sensitive to challenge and apoptosis. We aim to deliver the siRNA, shown here in purple, which will block the mRNA for the itch protein. Itch won't be produced. Then within the tumour cells, because of the targeting nature of the delivery vehicle, you'll get a build-up of p73, resensitising the cell to chemotherapy or radiation, and potentially more targeted therapies. And then the cell dies. This here shows the delivery vehicle, protein protruding throughout the whole structure. The yellow is the lipid. Within the central core is the siRNA, the payload. As I said, this could be DNA, plasmid, mRNA, artificial chromosome. For oncology, this will be given as an injection, infusion. It enters the bloodstream. The vasculature around tumours is very leaky, immature, constantly rebuilding. For most of the other liposomes that other people have developed, that is as far as their targeting goes. It just relies on leaky vasculature. Because we have receptor recognition sequences on the delivery vehicle, it can bind to receptors, in this case the integrin receptors, and get actively taken up into the cell. And we have very nice in vitro and in vivo data showing that if we don't have these targeting sequences present, the take-up efficiency drops off dramatically. Once inside the cell, the particle starts to dissociate. This is partly the role of the liposome component of the nanoparticle. It helps the cell actually digest the particle, releasing the siRNA payload. And therefore, in this case, blocking itch ligase, but obviously it could be any gene you choose. This just shows a snapshot of some data of this lead product. We've also done this in a pancreatic cancer cell xenograft model. This is tumour growth. Vehicle alone, tumours grow to a large size. We treat with docetaxel, which is a standard chemotherapy for advanced prostate cancer. A small but non-significant decrease in tumour growth. Our product alone, ECP102, the red bar here, no effect, as we'd expect on its own. However, when we pre-treat with ECP102, in this case it was 3 tail vein injections, giving 24 and 48 and 72 hours prior to docetaxel treatment, we then hit with docetaxel, we now see a significant effect. So we're sensitising in vivo xenografts to chemotherapy. We have in vitro data as well showing we can sensitise to radiation therapy as well. So this is targeted gene engineering, and this really is one of the keys, because obviously with this we wouldn't want to raise p73 throughout the body. And this here, in this case, the payload is just a fluorescent siRNA, so we can trace where the particle's going. And in this case, it's a neuroblastoma tumour model grown on the flank of a mouse. A single tail vein injection, you can actually see the injection site fluorescing slightly there. And this really is one of the key slides for the LipTide vehicle. When we actually look, this is at 24 hours post-injection, there's fluorescence in the tumour, there's no fluorescence in any of the other major organs, and it's not shown on this slide, but the product doesn't cross the blood-brain barrier either. If this was just a standard liposome, which many of the other companies in this space are developing, most of it ends up in the liver, and that's why many of those companies have changed their strategy now to focus on liver disease. We also now have got more data on this. 4 hours after injection, most of it is in the tumour. There's a little bit in the lungs, as you'd expect. You're giving it intravenous injection, so the blood will go to the lungs first. And at 48 hours, you still see it all in the tumour, not in the other organs. And we have 2 more products which as we merge with this company from UCL. One is a neuroblastoma product, and one has potential in cystic fibrosis. These are at a very early stage, potential for us to develop as we bring funds in. This just gives a hint of the data. This is a neuroblastoma tumour growth model. The top black line here on the left-hand graph here is tumour growth, vehicle treated. The blue and red, the mouse have been treated simply with LipTide containing an siRNA against an oncogene called MYC-N, which is overexpressed in neuroblastoma. By delivering siRNA against MYC-N, we flatline tumour growth. And in this case, the only difference between the blue and red lines is one's a positively charged LipTide particle, one's a negatively charged LipTide particle. And again, quickly, this was a single dose given as a nebulized administration to mini pigs. And in this case, rather than targeting integrin receptors, the targeting sequence was changed to target ICAM1, which phage display studies has been shown to be overexpressed in airway epithelial cells. And in this case, all that was delivered was purely a marker, a plasmid for the gene beta-galactosidase, which can easily be stained for with subsequent immunohistochemistry. 3 days after dosing, the animals were cold, immunohistochemistry was performed, and the brown stain here is showing where this gene is being expressed, which is in the airway epithelial cells, which would be your target cells if you're looking at a gene therapy product for cystic fibrosis. Importantly, when they looked at inflammatory markers, there's no inflammation. Because one of the issues, both with liposomes and with viral particles, is an immune response, which if you're looking to treat cystic fibrosis, obviously would be a disaster. And the options that you could put in here, we've got in vivo siRNA studies. You can knock out genes that exacerbate the symptoms, such as a sodium channel called ENaC. And also, we could license this to companies who do gene editing, looking to repair the CFTR gene, the gene that's mutated in cystic fibrosis. Very briefly, just talk a bit more about the company. Our chairman is Paul Murray, who comes from a private equity background, originally with Deutsche Bank. He's also quite famous in Scotland because he's also the chairman of Rangers Football Club. Our CEO is Alan Walker. He has 47 years experience at Big Pharma, was a vice president of Warner Lambert and Pfizer. More recently, he's actually built companies successfully, returning 30 times in 2.5 years to investors in his last company. I'm the CSO, Simon Newman. I've over nearly 20 years, actually, I calculated now, drug discovery experience and involved in spin-out companies from academia. And we're looking to fill those remaining positions as we go forward. I think one of the strengths of the company, actually, is the seniority and level of our scientific advisory board. And I had first-hand experience this the other week when I was interrogated by them. We have, it's chaired by Professor Alistair Strachan, who was biotherapeutics at Pfizer. We have Professor David Harrison, who's Director of Laboratory Medicine up in Edinburgh and works very closely with us, is also on our board. We have Dr. Frank Fields here, who's ex-head of global development for AstraZeneca. Professor Alex Mullen at the University of Strathclyde is helping us understand the technology from UCL more, and he's an expert around liposomes and also for the scale-up work. Professor Tim Hammond was one of the leading people at AstraZeneca on their safety panel for first-in-man study, so he's advising us around our clinical program. And we've also actually now, we have provisional agreement with Professor Gordon Jason at the Christie to lead our phase 1 trial. How am I doing for time? 2 minutes, okay. This is just a brief video here showing the potential in gene editing. So rather than delivering siRNA, we're delivering DNA for the key enzyme, Cas9, involved in gene editing, and the DNA also encodes the guide siRNA. The yellow was the correct DNA sequence. This could be given via injection, as shown in this animation, or it could be given via inhalation for cystic fibrosis products. And again, in this case, we're targeting the ICAM1 receptors, if this was cystic fibrosis, and it's taken into the cell. The one difference when delivering a DNA payload is the efficiency drops off slightly because the DNA is released in the cytoplasm, and then as with all gene therapy, you rely on it being taken into the nucleus through the pores in the nuclei to then get gene expression. But we've demonstrated this in vitro quite nicely, actually, that we can deliver the tools for gene editing and we can correct gene mutations in vitro using this delivery particle. So based on our experience of it, there's no reason why it shouldn't therefore work in vivo, basically, this delivery technology. Right, so that's it. I'll skip over this for the sake of time. The key things here now is we're looking to raise £7 million. This will get us as far as completing our toxicology program, which in this case would be single species monkeys. Because it's an siRNA-based product, we need to sequence homology. We can't actually go and use dogs and rats because their itch genes are very different. This would also cover us buying the UCL company spin-out, the UCL spin-out company, which we're then going to work with them going forward, but that's a £3.5 million cost associated with purchasing that company. and it would allow us to start our phase 1 study, which I say we're looking for April 2018, and we'll get as far as acquiring the initial safety data. And going forward, and we have a detailed report from Pharma Ventures if anyone's interested, they believe we can do about 1 licensing deal every year for the next 5 years, they believe. So cautious interpretation of that, we can actually look to start raising money based on the delivery vehicle quite quickly. And one of the key things we're bringing to this UCL company, which we were their only customer, is marketing expertise. Brilliant academic, they formed a company, did nothing with it. They've got a great product there, which we can validate with our lead product, and we're already putting a marketing brochure together with them. We're in quite advanced talks with the major pharma who are very interested in this technology, having previously had licensing deals with some of the other companies in this space, and I think been disappointed by their results. And I should also add the Scottish Government have agreed to put £2 million in as our cornerstone investor already. So we're well on the way, and it's a really exciting opportunity. And it's just me here today. Thank you.
Any questions for Simon?
In general, what's the format? So if I have like 6 questions, do I ask them now or do I wait for a one-on-one or?
We've got about 3 minutes for each presenters for questions, and there's a mic there, so please just fire away.
Okay, first, you block the ubiquitin pathway. Do you kill just the ubiquitination of P73, or you're killing a ubiquitin pathway in a more widespread way?
There's about 600 different ubiquitin ligases, so they're very highly specific. And obviously there's a big company called Mission working on small molecules for it, which is proving quite challenging. This itch ligase, its primary target does seem to be P73. There is some evidence in the literature that it is also involved in downregulating some other transcription factors which are involved in cancer, and actually if they were to become upregulated would not necessarily be a bad thing. But it is fairly specific. You know, they are a bit like the tyrosine kinases, a huge family with specific reactions.
And I trust you do have data that shows that you're not inhibiting P53.
We do have data. I mean, most of the studies have been carried out in cell lines where P53 is either mutated or inactivated. So I think in the one study
if you put it in a tumor that expresses P53, do you knock P53 down?
In the one study from UCL that I've seen where we've looked at just done protein expression of P53 as well, we've seen no change.
Okay. Does your particle enter the nucleus?
Our particle doesn't, no. So it'll break down in the cytoplasm. So if you're delivering a DNA-based product, you then rely on the passive uptake through the nuclear pores and nucleic acids into the nucleus. But we have shown, like, whether it be plasmid, artificial chromosomes or gene editing, we can get expression when we deliver DNA.
Okay,
I'm happy to catch you up later on a one-to-one if you want to.